Hello and welcome back to the Better Brain Fitness podcast. My name is Tommy Wood and I am joined as ever by my expressive co-host, Dr. Josh Turknet. Say hello, Josh.
Hello, Tommy.
Today we're going to talk about a super interesting paper that Josh found, and Josh is going to give us the rundown. It was published in PLOS One, sort of towards the end of last year, called "A Study of RNA Splicing and Protein Expression in the Living Human Brain." Josh, tell us all about this paper.
Yeah, so I titled this post for our Substack people that an existential crisis for Alzheimer's research, which is sounds hyperbolic, but I don't know. You be the judge. So, all right. So I do think this is a bombshell paper in a lot of ways. It kind of calls into question a large body of Alzheimer's research, and may also in part explain why the efforts to find a pharmaceutical treatment have been disappointing. So as background for this, there's an assumption that Alzheimer's research has been based on, or at least the bulk of it, which is that what you see in post-mortem brain tissue or dead tissue is what you also see in living tissue. And a lot of research in recent decades has centered around creating a model of how the pathological findings that are seen in the brains of those with Alzheimer's, things like amyloid plaques and neurofibrillary tangles, how those things form. And that model has been the basis for pharmaceutical research. And that involves these detailed molecular analyses of the tissue of the brains of people with Alzheimer's. And importantly, most of that tissue is tissue that was collected after they died. And the assumption there is that what is seen in that dead tissue is what is seen in living tissue. So last year, some people decided to look to see whether that assumption was correct. And so this paper was published in PLOS One last October 2025 under the inauspicious title of A Study of RNA Splicing and Protein Expression in the Living Brain. And what they did was they took people who were undergoing surgery for deep brain stimulation, so where they had to open their skulls and they had brain exposed, and they took a small sample of brain tissue during that surgery, and they had 275 subjects. That's pretty impressive. Mm-hmm. Then they compared that tissue head-to-head with post-mortem samples that were matched for age, sex, and tissue size. And then they performed detailed molecular analysis of that tissue. So they looked at gene expression. So what they specifically looked at was the expression levels of RNA, which is the transcript of DNA, part of the protein-making process. So the RNA levels tell you which genes are being transcribed or which genes are being active in the cells in those tissues. They also looked at the how the RNA was being spliced into its functional form, which determines kind of which version of the proteins will get made. And they also measured protein levels directly. And what they're trying to do with all of this is to figure out what the cells of the brain are doing, what genes are being turned on, what proteins are being produced, and then how that translates into function at the cellular level. So what they found, in short, was that the dead tissue did not look at all like the living tissue. About 74% of the primary RNA transcripts, about 70% of mature RNA transcripts, which is after they've been spliced together, and then 61% of proteins showed significantly different levels between the living and the postmortem tissue. And all in all, 95% of the transcripts they looked at were different from living to postmortem tissue in at least one way, whether that was at the expression level, the splicing rate, or both. And they also found that patterns of co-expression between RNA and proteins were fundamentally altered. So those are these correlations across molecules that kind of speak to the broader functional regulation of the cells, and sort of indicating that the differences that are seen are at the regulatory level, which is kind of even more significant. So all of this suggests, at least, that a large part of what we're seeing in dead tissue is the molecular signature of dying, which researchers hadn't realized. So why does that matter? I think it's probably apparent why that might be a big deal, but I'll give an analogy to maybe highlight the significance. So essentially, what researchers doing when they're looking at brain tissue to study something like Alzheimer's is they're trying to piece together, what happened, what caused the disease from a few clues. So very much like a detective solving a crime. So imagine there's a murder, it's been committed in a home, and investigators arrive at the crime scene. But between the crime and the arrival, the home was flooded. So water has moved the body from one room to another. It carried away the murder weapon and brought in a gun from somewhere else. Creates new stain patterns on the walls, shift the furniture around, scrambles everything. But when the detectives walk in, everything still looks like a crime scene, and there's no evidence that there was a flood. So it seems like you can still piece together a story of what happens. The problem is that's the story of the flood rather than the story of the crime. And that's what this study is suggesting is happening with the postmortem brain, that death isn't just degrading the evidence, sort of just, like it would getting a little bit dusty, but it reorganizes the entire molecular landscape. And it looks like there's still a story to unpack, but that story is telling you about something very different than the thing that you're interested in, which, is arguably worse than having no evidence. So just pursuing one red herring after another. We've said many times before that, one of the big limitations of Alzheimer's research in general, and I think a blind spot, is that the entire model is based on pathological findings, which has all sorts of limitations, and are only helpful if the functional deficits that are caused by the disease are leaving a clear trace. But that's even before this study came out, I think, which makes this scenario even worse. So needless to say, this, I think, does create a bit of an existential crisis for the mainstream approach and to research and the prevailing model of Alzheimer's. And as I mentioned, I think it's almost surely another key reason why the therapeutic efforts to find a drug treatment have been so disappointing. So that's my read on this. Tommy, what are your thoughts?
Yeah, thanks for that. And this might be the first time we're on the podcast we're going to disagree a little bit.
Okay.
Which I think is kind of fun. Because absolutely understand that interpretation. And my bias is these are some techniques that we use in my own lab. And I don't think, and we do it on dead tissue, usually from animal models, but we do do some stuff with human samples as well. And I don't think that I'm having that much of an existential crisis, but I think that the context is super critical. So when you think about this study in particular, I will say, right here, we're talking about it through the Alzheimer's kind of dementia lens, although most of these samples, both living and dead, came from people with Parkinson's disease. But there's no reason that those things shouldn't translate across, right? Because the fundamental idea is what's seen in living tissue, translated when we look at it in post-mortem tissue. So they actually did a really good job of trying to make sure that there weren't other reasons why they were seeing these differences. So this is actually a follow-up paper. Previously, they looked just at gene expression. It was a paper published in Molecular Psychiatry a couple of years ago. And they looked at things like post-mortem interval. So like the time between death and when the tissue is collected, or it is, even though the human, right, the heart has stopped beating, they are technically dead as we would consider them, right? There's still stuff going on in those cells, right? Proteins are still active, right? There's still some aspects of metabolism going on, right? So things can shift. But they found that post-mortem interval didn't seem to explain some of the differences. They also looked at a whole bunch of different things related to the fact that, right, we're not looking at living and dead tissue from the same people. So like, are there other factors related to their diagnoses or health or other things that might be explaining it? That didn't seem to be the case. So there was absolutely like some signature of dead tissue versus live tissue. They also looked at, you know, batch effects from sequencing, which can and batch effects of looking at the pathology, which can have a massive effect. Actually, batch effects for running proteomics and that kind of stuff can also have an effect. This is, you know, we've done some studies where things were sent to the core facility that does the analyses in 3 different batches, and between the different batches they changed how they did the method slightly, and all of a sudden it becomes almost impossible to compare across batches. So like, they kind of looked at all of that, and that didn't seem to be the case. One thing that I found particularly interesting was that when they looked at expression networks or proteins and genes that they thought, you know, sort of like core like fundamental parts of the Parkinson's disease process, they were much better represented in the living tissue than they were in the dead tissue. So like, that's where you could start to say, oh, hang on a second, like you're kind of missing these key things that we think we would expect to see in the tissue in the post-mortem versus the living tissue. However, when you're comparing like for like, right? So when you're comparing within post-mortem tissue or within living tissue, right? There's a lot more similarities by, you know, the status of the tissue when you collected it. But that doesn't mean that you can't find useful signatures within that subgrouping. So you couldn't find useful signatures within the post-mortem tissue. So like, when we do an injury model in the lab, right, we then have post-mortem tissue that we're looking at, and we can still see signatures of an injury, say compared to a control group, or we can see signatures associated with a treatment relative to an untreated injury. So and that makes sense in the context of the injury that's been generated, or the function of the treatment that's been applied. So I don't think that this kind of information like completely invalidates the process. We just have to acknowledge that a lot of what the signature you might pick up is driven by the fact that this person died before the tissue was collected, and that therefore the likelihood that any findings will translate to treating a living person are decreased. So I don't think that it like completely sort of overturns everything, but it allows us maybe to put findings into better context, and again, just be a little bit, you have a little bit less hubris about this idea that we can suddenly figure out everything out by just like studying dead brains and sequencing and then measuring the proteins, and we'll figure it out in that sort of reductionist manner. I think at this point we've appreciated that approach is probably going to fail many more times than it's going to succeed. Right. So like, this very much fits with how I would think about using these techniques, but that's, you know, from the kind of the viewpoint that of the things that we've already discussed. But you know, I will still say that there's some potential utility in doing these things, even with post-mortem tissue. We just have to, you know, be honest about the fact that it's not completely, or you know, it's not mostly reflecting what happened in that person's brain when they're alive. And even then, right, we know that we can't at this moment in time, or probably ever, measure all the genes and proteins and that kind of stuff and like have that tell us something really meaningful about how that person lived and interacted with the world, right? Those are two very, very different things, and like that's an even bigger question that sort of comes into play here. So super interesting paper. I'm really glad you found it. I think that this is important for neuroscientists really to kind of take into account when they're thinking about these things, because I'm not sure we do enough of that. But I'm not going to like throw these techniques out of my lab because of this paper, because I still think there's some utility there.
Right. So yeah, I think that is definitely a reasonable take, and I think that that doesn't mean that there's not room for post-mortem analysis at all.
Yeah.
I think, to me, the concerning thing is one, what do you do with the data that you've been looking at all this time and basing all this stuff on, right? Can you discern the signal from the noise in that, at this point in time? And then two, like, how does this, if you are going to continue to use post-mortem tissue for constructing models and trying to figure out what's going on, how do you do that in a way that's not, that's actually finding the signal? And I think that seems to be like a pretty important question to try to figure out. So, ideally you'd have more. It's tricky, and this obviously, like, be nice if this is replicated, but it's a hard question to answer. One challenge being living, just finding living, living versus postmortem tissue in the same person. Do you know, has there been animal research to look at this question? And if not, can you do it?
That is a great question. I don't know if that's been done. It would certainly be possible. You would just have to do you can do a small craniotomy. You can take a small section of tissue, and then sometime later you can look at it post-mortem in the same animal. I haven't seen that, and reading the paper, because they're fairly recent. I feel like if that had been done in animal studies, that they
would have put it in.
They would have put it in there as like some background or something, and I didn't see it. So worth looking at, and certainly something that we could do, actually, because we do the kind of surgeries that would allow us to take those samples,
right,
and then allow the animal to survive afterwards. So just like they did with the DBS, the deep brain stimulation. Right. You just take a small amount of brain. They took a small amount of brain where normally you just cauterize that brain to insert the probe, and here they just took a section out instead. I thought that was quite a neat way of doing it. So you can remove small sections of like the prefrontal cortex without massively impairing function. So it is possible. Something to think about. Maybe that's a new grant proposal that we can write.
I like it. I mean, yeah, I feel like it needs to be done. And I don't know if maybe these guys are working on it, but yeah, great. So I think we're at consensus. I don't really think we had that much faith in the model already, but I don't think this augments it in any way. I think it calls it into further question, maybe by varying degrees. But yeah, I don't think that this, you don't need to throw the baby out with the bathwater. That there's still going to be a role for postmortem tissue analysis, but getting smarter about it is probably going to be something important in light of these findings. So
absolutely,
we'll leave that to you.
We'll try and move that slowly in the right direction.
Exactly.
We'll see. Yeah.
All right. Well, thank you guys for listening. If y'all have any questions related to this topic or anything else in the realm of brain health and fitness, feel free to send it our way. You can leave it on a comment on Substack, or you can click the link in the podcast description, or go to brainjo.academy/questions, and we can find it any of those ways. All right. Thanks, Tommy.
Thank you, Josh, and thanks everybody for listening.
All right. Bye-bye.
Bye.